Recording. Hmm. All right. So here's a whiteboard. So today, we'll discuss questions with I would say hard, moderate to hard questions, right? So in last, I think this the topic of nucleic acid that we have taken, and from there I have discussed like some easy questions from that portion. So today I will go to start with some complex one as well. All right. So this is how we'll try to practice the maximum questions and then we'll go for the next topic. All right.
With the nucleic acid topic.
Yes, because nucleic acid I have just discussed with you not
Yeah,
all
which
questions.
topic? Nucleic acid one, yeah.
Yes, because the transcription and translation portion was bit remaining from there. So we'll try to get questions from there as well, okay? Yeah.
Okay.
What's your comment? Are you gonna go for the next
Yeah.
topic?
Yes.
Yeah, that's why I just want to
No,
complete the question.
no. We can.
All right. So I just want to have the maximum questions from there and then we go for the nucleic acid. I've designed my worksheets based on a simple, moderate, and then hard questions. Nucleic acid. And show nucleic acid. A-level biology. Yes, we or not. This is not the thing. I just want the questions from there. Nucleic acid and today I think take questions from medium soil sort of thing. Okay. And then we go for the complex one as well. Okay, Abdul, are you ready for this? Are you ready?
Yes.
Okay. So somewhere you're feeling I'm not you're not able to
Yeah. Yeah.
understand. So you can ask me as well. Okay. So the first question it's a medium level of question that I'm going to face as compared to the questions that we have discussed yesterday. They were bits of simple one. So few of the medium and few hard questions that I will discuss in today. So here I'm going to start with you. If I can paste it here. Oh yeah, it's pasted. So here's a question. Question is, I have started recording. Okay. So question is just in front of you. You can see figure 16.1 shows the structure of ATP. What you need to do is name the circled component in figure 16.1. Name the type of reaction that occurs when ATP is converted to ADP. A teacher told his student that the human body makes the equivalent of its own mass in ATP every day. Explain why at the end of the day, only a small proportion of the student's mass was ATP. So this is what we are trying to handle the question here. So let's start from the first one. Name the circled component.
Yeah. So the circle component, it is the nitrogenous base.
Hmm.
Yeah.
ATP. ATP. Nitrogen is base, but we have to specify here because that's not something like we cannot give it a general name. It's a nitrogenous base, but which nitrogenous base? This is also important here because we are talking about ATP right now.
I
So
don't.
it
I don't. I don't know. I don't know. See.
just encircle thing, not nothing else. I'm not talking about adenosine triphosphate.
Yeah.
It's the full name of it, right? But here I'm just talking about
Yeah.
this encircled part that you have to name it.
Adenine.
Yes, it's
Adenine.
adenine. This is nitrogenous base in ATP, right?
Yeah. So the reaction that occurs when the ATP is converted to ATP
Yes,
It
which
is
reaction is there?
hydrolysis
Which
reaction.
reaction? Yes, you're a bit. Yes, you are correct in it. So you can write this name there. What's that? Hydrolysis. This is what you said.
Yeah,
Okay, and you know what is hydrolysis? Condensation, hydrolysis. These are two uh
yeah.
reactions that we used to see in such type of chemical reactions in biological molecules. So what happens in this case in hydrolysis? What you did here?
The water is the water is used up in this
Yes,
reaction to
it's
break the
actually
bonds.
exactly. So ATP is converted into ADP by addition of water, right? That's why I'm saying it's a hydrolysis. Are you getting?
Yeah.
All right, that's good. Follow, sir.
So I can
Yeah. Nobody's there, sir. Yeah. Okay, okay. Abdul, yeah, please carry on.
explain why at the end of the day only small
Yeah.
portion of the student's mass was ATP because rest of the ATP was used up
Yeah,
in
yeah.
entire day.
S- Um, hmm. ADP is rapidly broken down to ADP to release energy, right? Okay. Hmm?
I'm saying the rest of the ATP was used uh,
You
in
know,
the
it's
entire day.
actually the problem. What is the main thing that we need to talk about? Because it's a very unstable molecule, right? So ATP rapidly broken down to ADP to release energy, right? This is one thing that you should
Yeah.
keep in mind. And as we know, like it is then regenerated from ADP and phosphate. So I would say like it is recycled many times per day. So at the end of the day, once you will see like only ATP that would be small in mass. That's simple because this process is carried out, or like this process is always carried out in the body of an organism. And once it is produced, the other way it's going to be broken down. So this is a cyclic process. Okay. So that's why we can't get the enough amount of ATP there.
ATP rapidly broken down into ADP.
Yes,
Uh,
to release energy. Because why this happened? Because we are doing some activities. So in order to that, we need some energy in the form of ATP. So ATP has to be broken down into ADP and inorganic phosphate, uh, because we need energy there. Are you getting this point or not,
yeah.
Abdul? Yeah. Then after you can say, then it is regenerated
For
from ADP and phosphate. The point is like it is recycled many times per day. This is the overall the I would say the crux of the discussion is it is recycled many times per day. So that's how we would say that the small proportion of the student's mass was AGP because that has to be converted into ADP and inorganic phosphate and energy that we really need if we are doing any sort of activity, right? Getting?
yeah. Yes.
All right, Abdul. So I hope uh, it's clear to you now.
So in mitochondria, basically the phosphate molecule joins to the ADP to make it ATP.
Yes, exactly. Because ADP and inorganic phosphate is always there as a component once it is present there. So it's going to form another. Definitely, it needs, you know, how it is produced. If you know about, do you know about the ATP production mechanism? Like we know about in respiration process, the glucose is broken down and that's going up to the glycolysis, the Krebs cycle, and electron transport chain, and the energy which is released from there due to electron movement from one protein to the other. I hope you're getting this point or not. Can you just recall that concept?
Yeah, when the bonds broke between ATP and phosphate to when it changes into ADP, so
Okay.
the release of energy is basically the energy which is produced.
Yes. Abdul, it's a complete process again. My focus is right now to come on the questions, because there's important thing, like what you need to do is whatever the topic that we are discussing. So have you revised all that or not, in nucleic
Yeah,
acid,
yeah,
like? Okay. So yes, in that case, there's an electron movement from high energy level to the low energy level. So that because of that, it's releasing energy. So that energy is actually responsible for combining a DP with inorganic phosphate. Are you getting this?
yeah, yeah.
Yeah, that's the thing. So that's why we're
So
saying ATP.
then it's like an immediate source of energy. So when the phosphate again leaves
Ex-
the ATP,
yes,
so
because
it will
yes, exactly, and the glucose is also there. Like the respiration process is also continued. Are you getting?
yeah,
So,
yes.
all right. You can give me this bag, sir, please. Sorry, Abdul. Just a moment. Oh, okay, Abdul. Sure, carry on, please. I just want to add my tab here. Okay. So, should I give you next question? Yeah, that's fine.
Yeah.
Should we move to the next? Let me read the last one. ATP rapidly broken down into ADP to release energy. Then it is regenerated from ADP and phosphate. Yeah, one point you need to add here, because it's of 3 mark, 2 mark question, but you need to add one more. Um, the point that we need to add here is, like,
It's a reversible process.
it is recycled many times per day, because he has talked about the day. Mass was ATP, like, small proportion of students mass was ATP at the end of the day. So that's the point, that it is recycled many times per day. That's what you need to add here. Great.
Yeah, done.
Then after we can go for the next question. DNA can be obtained from the variety of plants and animal cells. And what we have here, based on this, there's a question: state the purpose of statement. It's a 5 mark question that I'm going to give you now. Oh no, explain why at the end of the day only a small proportion of it. Then why it is only of 2 marks question? It should be a 4 mark. Oh, yeah, four is overall, I think. Yeah, 4 marks are overall, like two, 4. Okay. So now I'm going towards another question that's a 5 mark question for you, and just need to read all of this, and then after we'll discuss. And the question related to this here, I have to paste for you, and from my worksheet. Mm, copy. Okay, just read out this. That's for you. And meanwhile, I'm putting one more for you, like this. These are the questions which are related to the above mentioned discussion. Just read out carefully. It's a 5 mark question. You have to answer one by one all of these questions. 1 mark each question, right? So
Yeah.
meanwhile, I can do one thing. Abdul, wherever you are confused, like you think like this question is are the background concept I'm not able to understand, I'm confused, so you can discuss with me. So there I will elaborate as well for you, okay?
Okay.
Oh my goodness, where's wire? Oh yes, it's here. Okay, fine. Abdul, you can read out for me as well. Try to discuss question. I mean, not in a passive way. You should read out the question completely and try to dissect it properly, like what we can do here.
State the purpose of step one. So it
Hmm.
is grind a leaf to fine pulp using a pestle or mortar. Uh, uh,
DNA can be obtained from the variety of plant animals. A group of students try to purify some DNA from leek cells using the following method. They decided that exact volumes were not necessary. Grind a leek leaf to find a pulp using a pestle and mortar. Add salt and cold water and mix again for at least 10 seconds. Add protease enzyme and mix again for at least 10 seconds. Filter the liquid into test tube and stand for at least 10 minutes. Tilt the test tube and gently pour in ice cold ethanol. A white layer of DNA forms between the sample and the ethanol. Extract the white layer carefully using a glass rod. State the purpose of step 1. State the purpose of step 1. What is the purpose of step 1 here?
it is really part of my specification too, because like in my textbook I didn't read anything
You
about
didn't
this
talk about
experiment.
the DNA extraction.
Yeah, it was with
It's a
the
skill.
the ultra. It
It's a
was
skill
through
based
the ultra
question.
centrifugal for the
Yes.
Melson and the Star experiment.
Yes, exactly. So there, this is actually why do we need to grind things at the first stage? That's the point we need to figure out.
To
Like
extract the DNA.
grind the leek leaf to a fine pulp using pestle and mortar. So what is somewhere you need to apply the concept, whatever the concept that you have related to this. So definitely, as you said. So yes, we are there to extract the DNA. Our main purpose is that we are going to extract that. So just why, okay, state the purpose of step 1. Um, what we want to do here. We are using that in order to grind that. Grinding is actually that's help you to separate the cells from there. You can actually, what you really want to do is like you want to extract, you want to get cells from that part of it. This is one thing. Definitely, we are grinding anything. So it means we are converting that into small pieces, into a fine pieces. And definitely then after so that you can apply the protease enzyme. Actually, you want to do what? You want to increase the surface area of that. Once you are dividing those substances, sorry, that leaf into a smaller pieces, what you really want to do, because just after this you can see we are applying enzyme there, and enzyme can act efficiently where it has a large surface area. Are you getting? So that's
Yeah.
why in order to have better efficiency of enzymes, this is another purpose that we can add here. We are actually trying to convert that leaf into a smaller pieces in order to increase the surface area. Okay.
So we we
Hmm.
make fine pieces to increase the surface area.
Yes.
To increase the surface area to volume ratio, isn't it?
Exactly. This is what I'm saying. Surface area to volume ratio will also increase.
I think we will write the enzyme concept on the second question, isn't it?
What's about the second?
Or I will
What's the second question here? Just read out.
why a protease enzyme added in step 3 is needed to purify DNA.
Is suggest why a protease enzyme added in step 3 is needed to purify DNA. Yes, because and for this purpose, what the concept that you must have? Histone
The
proteins.
see yes,
I mean the
histone proteins.
because DNA itself is not only the DNA that has proteins as well. So if we want
Yeah.
to get the DNA, so we need to digest those proteins which are associated with DNA, right? So that's why, because we want to separate those proteins or digest those proteins in order to get the DNA from there.
Yeah, done.
Done.
Yeah.
All of the questions or
Just
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 one?
there's two. So now third is the student considered using pineapple juice as a source of protease enzyme. Suggest why this wouldn't be an appropriate source of protease when attempting to produce a pure sample of leak DNA.
Hmm.
So it can dilute the sample of DNA.
Mm-hmm. Just a moment.
Is it?
Okay. The next question is the
So
student
just
considered using a pineapple juice as a source of protease enzyme. Suggest why this would not be an appropriate source of protease when attempting to produce a pure sample of leak DNA. Hmm.
this will contaminate the sample of leak DNA.
Because he's taking pineapple juice. Yes,
Yeah.
exactly. Because whenever we want to extract the DNA, we have to use the pure protease, not with any other. For example, if that enzyme is present in pineapple juice, so we can't use the pineapple juice there because there are many other contaminations which would be there in that case. So yes,
So
we always get protease enzymes separately in a vial. Okay, because it is available in the
yeah,
market as well. Once you're doing DNA separation, so we always use this, right? This is the molecular biology technique that we used to have. Getting?
yeah, yeah. I have written that it will contaminate the sample of DNA. So we need protease enzyme separately.
Separately. State one important step that the student had left out of their method.
Repeat the experiment at least 3 times.
State one important step that the student had left out of their method. Name the process described in step 6. No, that's is the question is related to, you would say, like, um which step, which important step that he left, that students left actually. The students considered, that's fine. State one important step that the students had left out of their method, right? So which step is there? Let's get back to the steps here. So what you can see,
Measuring the volume of the
mm,
liquid into a test tube.
how you can say, like, which one
From
is
step 4.
okay. Grind the leek leaf to fine pulp using a piston mortar. Add salt and cold water and mix again. Add protease enzyme and mix again for at least 10 seconds. Filter the liquid into test tube and stand for at least 10 minutes. Tilt the test tube and gently pour in ice cold ethanol. Okay, is there any step that students left? A white layer of DNA forms between the sample and the ethanol. Extract the white layer carefully using the glass rod. Glass rod. Okay, so this is the last step. So we need to find a step which is, what's the concept that you should need here? Definitely, you should know all the steps which are required for DNA extraction, right? That's what you
Actually, I
should
didn't
know
study
first.
this, but in my specification, the experiment was the Melson and the Star experiment, not this one. So that's why it's new thing.
Um, what's about your skill biology, like where you have the practical portion of your subject? Somewhere you have to, definitely this is a very basic molecular technique, like we used to give to our students, like where they must know how to extract DNA, right? So
Yeah.
the overall idea that must be, I think that should be to every student, like they should know how to extract DNA. There are few steps for DNA extraction. So you will, right now you are in AS,
Year
right?
one.
Year one.
Yeah.
So when you started this,
Last year.
so when you're going to
No, I
appear
have
in your
complete.
when you are going to appear in exam,
No, I have completed my year one. My year two is gonna start in September.
That's going to that's I mean, so A level going to start in September.
My first year is completed. Now
Okay.
second year, the A2 is going to start in September.
So then, okay. So are you not, I mean, prepare like practicing these questions from AS?
Yeah, I am. So, but this experiment was a bit new for me.
It's, let me tell you one thing, Abdul. You will also do this experiment in your A level exam as well, like where you have to do some practical portion of your exam. So this
Yeah,
is just a simple methodology by which we can extract DNA. And the steps which are here, I'm just giving you an overview on this, and then after you will get this idea how to extract DNA. Measles and Stahl's experiment is just there because that was actually the first experiment where you identified DNA somewhere, right? This is the
yeah,
method. For example, you are getting any of the samples, and your task would be, they will assign you this task, like find out the DNA or extract the DNA from this sample. For example, in this case,
yeah,
we have started the plant tissue. That's a plant leaves, right? So
yeah,
like, at the first stage, we always use the lysis buffer, like detergents to break down all the cell membranes and like that. That's why I've used salts to stabilize DNA and proteins, right? And
yeah,
buffer to maintain pH. Buffer is always used like tris HCl. So it's a complete process because I used to and give these instructions to my BS students. So there I have a lot of practice on this. So there we always use a buffer solution for this, right? Then after we have to remove the proteins and cell debris. For that purpose, we have used protease enzymes, protease K,
yeah,
like in order to digest proteins. And
yeah,
we are also using phenol chloroform extraction. Like then after you can go for the centrifuge to separate the debris. Okay, centrifugation is a process in which we always separate things based on density. Then there's a process that's called DNA precipitation, right? Where you need to add cold alcohol to the aqueous solution and DNA will precipitate there. That will be settled down in the base of any tube that you have taken. Okay. And then what we
yeah,
always do, like we often keep that in -20 degree or even the colder for uh, better yield. Like we always keep that in the low temperature so that we can get better yield from that DNA. This is just an overall idea, right? You don't need to get into. So the question is there any step that in which they have washed the DNA? So
no,
so it means like this is a step, I would say, like that is missing here. Filter the liquid into the test tube and stand for at least 10 minutes. Tilt the test tube and gently pour in ice cold ethanol. A white layer of DNA forms between the sample and the ethanol. Extract the white layer carefully and using the glass rod. So yes, there is a step which is missing here, Abdul. That is that they didn't wash that DNA at 70 with 70% ethanol. Have they used ethanol?
yeah.
If they have used ethanol,
Eh,
so it means that's a washing process.
no, in the fifth step they have used it.
What? Ethanol. In fifth
Ice
step,
cold ethanol.
ice cold ethanol. That is actually to remove salt and impurities from there. It means that step is also included. We need to find out the step which is not involved here right now. So, is there the use of tea buffer or any nuclease free water to dissolve the DNA?
No.
And listen, Abdul, for this purpose, what you need to do is you need to go through all of these steps of DNA extraction, right?
Yeah, I will do. Yeah, I will
Okay,
do.
then after you will be able to understand this all. Otherwise, here what we have, name the process described in step 6. What was the step 6? A white layer of DNA forms between the sample and ethanol.
Yeah.
Yes, is this not DNA precipitation?
Yeah, these are DNA precipitate which we need to, which we are
Yes,
looking for.
yes. So this is DNA precipitation, right? In step 6, what is mentioned here. So you can add here. Overall, there are 5 questions, but again, I would say just go through this DNA extraction process because you will get this question in your skill-based exam as well. Okay, so I'm moving to now. The questions would be a bit hard for you because I've just taken questions like figure shows the structure of a small section of the DNA cell during transcription. What you need to do is identify Q, R, and S as indicated in figure. Explain how you reached your answer. So I hope now if you have an idea, because the previous questions were a bit easy questions, right? I'm going towards now the hard one. So here's a figure that I'm going to paste in front of you. Okay, this one. Abdul, can you look this? Is this visible to you?
Yes.
Figure 1 shows the structure of small section of DNA in a cell during transcription. All right,
Yeah.
go through this all, and then after I'll put some questions in front of you. Is this structure anything that you think like it's new for you?
No.
This is the common structure of DNA, right?
Yeah.
So what you need to do is like you have a 3-mark question here that I'm going to paste just after this diagram. Here it is. Identify the bases Q, R, and S as indicated in figure 1. Explain how you reached your answer. And this is what you also explain it. That's also a challenge for you.
Yeah. So, uh, 1 second. Q R S and
You just need to identify the bases Q, R, and S in figure. And definitely, once if you go in that, so you need to look at the base pairing rules and chemical structure of each base, right? This is what you need to
yeah.
go for that.
So firstly, Q is gonna be adenine because it is bind with the thymine, so it is definitely gonna be adenine.
All right, that's good. DNA base pairing rules. Q is
Yeah.
what? Q is paired with thymine. So thymine pairs only with adenine.
Yeah.
Good. So adenine has how many rings?
2.
2 rings. And how many hydrogen bonds it's going to form?
Uh, 2.
2 bonds. 2 hydrogen bonds, right? So therefore, Q is what? It's adenine, right? Good.
Yeah.
Go for the next one. What's R here? Identify R.
Yeah. The R is going to be uh cyt cytosine.
It's cytosine. R is paired. Cytosine it is itself there that is paired with R.
Yeah, S and R. Like we have to find both. So one of them is S, guanine, and one of them is cytosine.
But you had to first find the R. Then after we'll go for S.
Yeah, R is guanine
R is governing. R is paired with
because it's
Hmm.
it's
Because it
two.
is it is
It have 2 rings.
2 rings.
Like the
Cytosine. Yeah. Okay. Uh, Governing is purine. Yeah, it's a double ring. Okay. And what's about cytosine? How many rings in there in case of cytosine? That's in front of you.
one,
1 ring, single ring. And how many hydrogen bonds are there?
3.
3 hydrogen bonds. Okay. So therefore, R is governing here. So then after you can go for S, as you already you know about it. S is paired with
Cytosine.
S is paired with cytosine or
Oh,
governing.
sorry. So,
Yeah,
guanine.
it's for governing. Governing. So governing pairs with cytosine, which has a single ring and 3 hydrogen bonds. So therefore, S is cytosine. Okay.
So I'm gonna write each my reason for the answer. Okay.
The reason for the answer is what you have already that I have already
Yeah, yeah,
explained with you. Okay, so
yeah,
this is
yeah.
what you know. First, you need to identify, and then after you need to write the reason how you have identified. So there you need to tell like the other one pair base is this, and these are the hydrogen bonds which must be there in case of any specific nitrogen bases, right? Getting Abdul?
Yeah.
Good. And how many marks are here in this case?
You
Three?
told me, I think three. Yeah.
Yes, it's a 3 mark question. Exactly. So you need it means like um 1.5 for identification and 1.5 for reasons as per mark scheme. Okay, fine.
Okay.
Done, Abdul? Are you doing?
Hmm.
Oh yes, you're
Yeah,
writing.
yeah,
Okay,
yeah.
okay. Okay, just write.
Yeah, done.
Uh Q is added in because it has 200 bonds and joined with thymine. Okay. Um what you need to do here is you can also talk about the number of rings as well somewhere like yeah, so that's for
Uh,
identification.
yeah, I can include in the first statement, but I already included in the second as well, but I can include in the first one as well.
Yeah. Okay. Second is included, yeah. As it has double ring structure and S is cytosine because it is bind with guanine. Yeah, that's fine. And because it has two hydrogen bonds and join. Okay, fine. That's good. So I'm going to have one more question for you, like this one. A section of DNA molecule contain 124 nucleotides. Calculate the maximum number of hydrogen bonds that could be present in this section of DNA. Show your working. Yes, you have to show your working. It's a 4 mark question, right? So as per mark scheme, you need to add 4 points like understand the structure of DNA. As you know, DNA is double standard and each base pair involves two nucleotides. So the number of base pairs,
Yeah, so number of base pair is going to be uh 62.
um, yes, overall 124 by 2. No, once you will,
Yeah.
I mean 124 divided by 2. Then after you'll get 62 base pairs. Yeah, that's fine. So base pairing and hydrogen bonds. That's what you need to talk about. So adenine pairs with thymine, it's of 2 hydrogen bonds, and guanine pairs with cytosine, you have 3 hydrogen bonds. So maximum number of hydrogen bonds, what you need to
Yeah,
do is,
we will do 1 second. So what we will do is like we have 31 with three hydrogen bonds and 31 with two hydrogen bonds.
and what will be the overall calculation
1 second.
to get the maximum? Is this not assume all 62 base pairs are GC pairs? Is for assumption
So yeah, it's 155 total.
155. What's
The
like
maximum
if you take
time.
just 62 multiply 3 hydrogen bonds, suppose in one case. Yes, fine. So overall, um okay.
No, 62 times 3.
No,
Uh,
why are you not taking like 62 base pairs that you already have? So you assuming here like all base pairs
half of
are
them half of them
GC
have
pairs.
two hydrogen bonds. No, I'm assuming that half of them has GC and half of them has the AT.
You need to assume something like here. To get the maximum, you need to assume all 64 base pairs are GC or 80. Like this is what you can do in both of these. And in that case, what you need to do, and then after you multiply the total number of hydrogen bonds there. For example, you're taking GC, guanine cytosine. So as you know,
So I think we should take the bigger number 3 because GC because it said it is a bigger number so it can
yes,
give a maximum value. So it's gonna
maximum.
be 186.
Exactly, maximum. That's the point. This is what we are trying. It's a catch here. Like if you'll take just 80, so there will not get the maximum. Maximum would be in case when you take 62 that you have to multiply with three hydrogen bonds. Three are the maximum hydrogen bonds there in that case. So overall, you will get 186 hydrogen bonds there. All right. So that's how you can go with this.
Yeah, I'm done.
62 base pairs are present in 124 nucleotide. Okay. And 62 multiplied 3, you'll get 186. That's what you need to work on. Okay, fine. Um,
Should I write that we are assuming all bonds have three hydrogen,
Yes,
all base
exactly.
pairs
This is
have
what
three.
I'm saying. Like you need to, there should not be any ambiguity here. Like examiner should, once is just looking at your question, so he should be able to understand everything from there. Because you can't put anything of I mean, you would say like in the code of examiner. Because he will not assume anything for you. Whatever you are doing here, try to write here because you have 4 mark question here. So it means like you need to talk about all of these things as well. Okay.
Yes, yes.
That's great. So then after we are going to have questions like ATP is the energy carrying molecule which fuels chemical reactions within the cell. Compare the structure of ATP with the structure of DNA nucleotide. I think it's a simple one, not mediums like it's not much simple, but approachable question. Like so, 3 mark question that I'm going to paste in front of you related to the basic concept of nucleic acid. Abdul, once if you feel some tiredness, so you can ask me like to get 2 to 3 minutes for break as well. But if you're fresh, okay, so you can continue. Otherwise, here's a question.
Yeah, it's fine.
Okay, so here's a question. ATP is the energy carrying molecule which fuels chemical reactions within the cell. Compare the structure of ATP with the structure of a DNA nucleotide.
Yeah.
Got it?
ATP contains
Mm-hmm.
three phosphate molecules,
Hmm. Good.
whereas
Hmm.
DNA only contains one phosphate molecule.
ATP has 3 phosphate groups, whereas DNA nucleotide has how many? Only
One.
one. Yeah. And
So,
the
and
sugar in ATP is ribose. In
yeah,
DNA, that's deoxyribose, right?
second, yeah, secondly,
And in HCP, you can see like there's only one nitrogenous base, one type of nitrogenous base.
yeah,
That's only adenine.
yeah,
However, in DNA, you have many other. Like these are some uh, differences that we need to talk about. Both ATP and DNA nucleotide contain although they have the commonalities only in the way like they have the phosphate group, the pentose sugar, and nitrogenous bases, right? So what we need to do is like secondly the sugar is ATP ribose, but DNA has deoxyribose sugar, yeah. And one more thing, like you can talk about the nitrogenous bases as well.
Yeah.
All right, good. ATP contains only one type of. Mhm. Okay, got it. Great.
That.
Okay, good. So, ATP contains three phosphate molecules, whereas DNA only contains one phosphate molecule. Second, the sugar in ATP is ribose, but DNA has deoxyribose sugar. Moreover, the ATP contains only one type of nitrogenous base, adenine. However, DNA has four types of nitrogenous bases, and you can also add in bracket like adenine, thymine, cytosine, guanine as well. In short, in just after nitrogenous base in bracket, you can say. Now, again, mathematical skill based question, and easiest one as well. Not easiest one, I would say, because I have taken the medium questions now. Here it is.
Yeah, I know
And
that one.
okay, good.
So the scientists
It's
analyzed the nuclear base within
it's the
DNA molecule
two
as
mark
a
question.
yeah. It was found that the 29% of the nucleotides in this DNA molecule contain nitrogenous base adenine. So it is like if it's 29% of adenine, so it will be 29% of the thymine as well.
Mm-hmm.
So in total, it will be 58% of the adenine
Mm-hmm. Good.
and thymine. So there will be 42% of the cytosine
Mm,
and the guanine. So 42 divided by 2 is going to
good.
be 20.5%.
42 by 2. It should be
41
20.
divided
It
by, yeah, sorry, 42 divided by 2 is going
yeah,
to be
it's simply
20 point,
42
yeah,
divided by 2. So you'll get,
yeah,
what's that? Add
21.
all, just try to solve it properly. Yeah, two mark question. Just
Uh,
add all of things here, like somewhere. I would
okay, okay.
say, yeah, where's that? Process of transcription.
Basically, I will write on my notebook, then I will paste it over here.
Hmm. Is it not possible for you to write here on the board?
The calculation will be difficult because it's only the option I have is text, like I can type it.
Uh, okay. So why not you
I will
using
just
properly?
paste.
Why you not using any like you have option here for pen as well? Like somewhere if you think like I need to use pen, so you can also use it. You have an option on the Zoom here on the one side.
Yeah, I tried, but I couldn't
Okay.
write it
Oh,
properly.
okay, fine, fine. That's good. That's fine. We'll go for next question, and that's a 5 mark question for you. And the question is, it's for the process of transcription. So you need to identify the molecule and describe its role during transcription, and then after we can go for to know their significance. Hmm. Here it is. Look at this question. The
Yeah,
process.
I'm just sending you the previous question. Just 1 second, 1 minute.
Okay, okay, fine. Mm-hmm.
Just yeah.
All right.
So yeah, the process of transcription is illustrated in figure 1. Identify the molecule X in figure 1 and describe its role in transcription.
Abdul, we have just completed the medium questions, right? And not even completed, but still we need some. I mean, in my worksheet, in medium questions, still we have a few of the questions remaining. Then after in hard questions, go to Maslow's and stall experiment. There we can talk about it, and then after need some
Yeah.
calculation as well. Gene sequencing is here. Like we can talk about this as well. One few questions from here and
So I think the X is the RNA polymerase,
RNA polymerase. Mm-hmm.
right?
RNA polymerase. Okay, what's about Y? Yeah, X is RNA polymerase, definitely. You know about it binds
Yeah,
to the DNA at the promoter
yeah,
site and like separate the two DNA strands.
yeah. And
And it's
Y
there.
is the strand Y is the antisense strand, which is also known as template
Hmm.
strand.
Yes,
Template.
exactly. Y is the template strand, right? So it's also called the antisense strand. As you know, it serves as a template for RNA polymerase to build a complementary messenger RNA strand, right? So yeah, the base pair of messenger RNA is complementary, like to this strand that is there already in the form of template strand. All right. So
Yeah.
yes, that's fine, Abdul. I think it's about 5 minutes remaining. So, but we can do. It's done. Now, yeah, you can add points here, but in tomorrow's class, I think we can talk about the hard questions. Today, I've discussed medium one with you. Okay, like
I will also prepare the cell cycle topic as well. We can do both things tomorrow.
tomorrow. So
The
it
yeah,
depends. Abdul, it depends. Like if you can cover all of these worksheets that I have, if I'll choose few of them as well. Like we'll take some questions from hard, and then we'll go for the cell cycle. But my point is like at least you should take, for example, these are the important topics definitely,
yeah,
and in cell cycle again that will go for the next week as well. So we'll try to complete this first, and it's hard portion. Still today I've discussed the medium one with you. Okay, tomorrow we'll discuss the hard questions from this topic, and then I'll talk about a few of the questions from the cell cycle as well, right? Okay,
yeah.
fine. So
Okay.
should we
And for
stop
the next
here?
lesson, what? for the next lesson, what we can do is that we are like the more than like three-quarters of the lesson we should spend on the hard questions. So you know what I mean?
Yeah, I mean, you wanna like we should spend time on the hard question, not to the even
More,
simple and the medium.
more,
More
yeah,
time to the hard, yeah. It means like in next, I'll take the hard questions from my worksheet and then we'll get back. If we'll have time, so we'll come back to the medium questions as well. It's a sequence like, once you started
yeah,
with the simple one, so you'll get more confidence to go for the medium, and we always take things based on human psychology. So like once you cover the simple, then we go for the medium and then for hard. All right. So but it's okay if I just get this point, like you are tackling questions easily in medium, then I'll give you the hard one, right? So it depends on you. If your response is quite positive in simple and medium, so I will pick few of the questions from there, and if I'll see like you are doing well, so then I'll take you to the hard one, right?
yeah. So
That
the significance of strand Y is the template strand uh, is the acts as the
Yes, it serves as a
acts,
template for the RNA polymerase. I have already explained, like
yeah,
to build the complementary messenger RNA strand, and the base sequence of messenger RNA is complementary to this strand, right? So this was
yeah,
actual thing that we have discussed. Got it?
yeah. To build a complementary DNA.
Yeah.
Yeah, done. Yes. So the role of the RNA polymerase is that uh, it joins the free it joins
The
the free
role of
nucleotides.
the role of RNA polymerase, that's there to
Yeah,
build a complementary messenger RNA strand. Its
yeah.
function
It
is to
joins
build
the
hmm,
it
the
joins the
base sequence of
complementary.
messenger is complementary to this strand, this template strand, and just it it except that the uracil replaces time, and this is what is in case of messenger RNA. So the base,
So it joins the complementary base pairing.
yes, it joins the build a complementary messenger RNA strand based on this template strand, and just by replacing, you would say like thiamine with uracil.
No, I'm talking about in the first question, identify molecule X and describe its role of transcription. So it's RNA
Um.
polymerase. It
Oh
joins the
yeah.
complementary.
All right. You're talking about the first one. Yeah. Bind to the DNA at the promoter site. This is what you can say. And yes, it separates the two DNA strands. I've already explained this before. And synthesize the complementary strand of messenger RNA by joining RNA nucleotides together, as you said. Okay. So it moves along the DNA template strand in the 3 to 5 direction, synthesizing messenger RNA in the 5 to 3 direction. It's moving on the DNA template strand from 3 to 5, but it's synthesizing messenger RNA from 5 to 3 direction, right? Getting this point,
Yes.
Abdul?
Yeah.
Okay. So this is how we should, yeah, you should add, and then we can continue this discussion for today. You must have the record of all these papers. How are you doing with that? Like,
Yeah, yeah. I say by the end of the by the end of the class, I always save the whiteboard in my file. So I have all the I have
okay, okay, fine, fine. That's good. That's good.
Mm.
Okay, let's conclude this for today, Abdul. Then ask to Abdullah to join the class.
Yeah, he will join in 2 minutes, like he was saying right now that he's
Okay.
doing something, so he will be.
Okay, fine, fine. All right.
Allah Hafiz.
Allah Hafiz. Take care. See you tomorrow.
Allah.